as a human being, you are living inside the media. Every day you're uploading things on Instagram, on Facebook, so you technically are part of the media. You are the media in a sense. And that is similar to bacteria in a sense, because the bacteria is living inside the media, and the media is a plural word for the medium. The medium is the environment in which the bacteria lives in. So the common medium is the agar. Agar is a transparent gel that is made up of seaweed. And you might be thinking, okay, so the bacteria is consuming agar, and that's right out the bat not true. Agar is just the environment the bacteria is going to live in. It's just like for us human beings who live on the earth, just like that, agar is the common medium that is used inside just inside normal labs. We have agar. That's what I'm trying to get at, is the bacteria is living on it. It's not consuming it. It's not using it to metabolize. It's the space it has. Plating is the process by which we put these bacteria onto a Petri dish. And the Petri dish is something you might have already seen in a bacteria lab. Sorry, a biolab, what I was trying to say. It's just a dish that you put the bacteria on with the common medium. And the medium is, like we just said, the environment. So this will allow the bacteria to reproduce over here, colonize, and that is the area, or the Petri dish is just a space where we can, or a dish, or just a tool we can use to allow the production of bacteria inside laboratories. You might have already heard the term wild type, and frankly, I was personally very confused back in high school when I was always listening to wild type. I never understood what it is until soon. And I mean, a wild type just refers to the fact that we have a bacteria or any kind of organism that displays all its characteristics from its species. And if we talked about a bacteria, E. coli, for example, the E. coli that we have inside the Petri dish is going to display all the characteristics that any other E. coli would. That's all it means by wild type. And if something is not the wild type, that means that it's missing some kind of characteristics compared to its original species. Its other members or its other species will have characteristics that it doesn't have. It's lacking something, or it might have an additional feature that the normal bacteria of its kind does not have. Now, of course, if we leave the bacteria on the right temperature with the nutrients inside the Petri dish, of course, it's going to colonize. And as it colonizes, you'll notice that there's some missing spots. There's some clear spots in the lawn. The lawn is just a dense growth of bacteria, and this is called the bacterial lawn. So this right here, for example, if we had a plague over here, that would be a missing spot. So that's that spot where these bacteria are dead. They're not growing on this spot, and that could be caused because a lack of nutrition. Maybe there's a toxin released in that spot over there. These bacteria can't grow. Something is limiting the bacteria from growing in that spot over there. So that's called a plague. The minimum medium is essentially just a medium, consists of glucose as its only energy source. And we just talked about it right now, that the agar as the normal medium. Of course, these bacteria need a energy source as well, and the most common energy source inside bacteria is glucose, and therefore the medium. If you ever hear the word minimum medium, that refers to the fact that the medium consists of glucose as its only energy source. Here's a picture from. I found this somehow. It was a miracle on my phone. This is the first picture that I took. I had approximately 25 attempts at least, and I was in my bio lab, and I, for the first time, saw bacteria under the microscope. It was a very long time ago. I was like, okay, look, you have these bacteria over here. This is what it looks like, a bacterial lawn. It's just a normal picture you'll see under the microscope. If you allow bacteria to replicate itself in a timely manner, you give it the time, the nutrients, the right environment, the right conditions, it will just allow the bacteria to grow. Now this brings us to doubling time. Doubling time is the amount of time required to double the bacteria. So we had a X, let's say we started off with 20 bacteria. How much time does it take to reach 40 bacteria? And that just means that how much time is it taking for the bacteria to double. And that is referred to as the doubling time. Of course, now there are limiting factors when it comes to bacteria. And one big limiting factor is the nutrients. The amount of nutrients and other factors such as space will determine how many bacteria can or colonize itself. How many times can they duplicate themselves? But these bacteria, so sometimes bacteria won't grow in a minimum media, and that means that if we just give it glucose and just has the agar, it's not going to grow, and that means that there is something missing. It could be either the energy source, that the bacteria is not able to break down the energy source, or it might be that there's a missing amino acid, like for example, might have a mutation, and therefore it needs a supplement. It needs a supplement amino acid for it to grow, and that is known as auxotroph. An auxotroph is essentially a bacteria that cannot grow in a minimum medium. It needs a supplement. Let's understand this through an example. A bacteria cannot grow without the presence of proline. Is this an autotroph? Well, the answer is no. It's not an autotroph. It's an auxotroph. So do not confuse these two words over here, these two terminologies. They're different terminologies. So auxotrophs are referring to bacteria that cannot grow in a minimum media. They need a supplement. So this will be denoted as pro-negative. So this proline, of course, proline is denoted as PRO. And we have negative over here, which just indicates that this bacteria is missing or needs proline to grow. If without proline, maybe there's an enzyme a mutation on the genetic information of the bacteria, and therefore they need proline to grow. It's minimizing, it just needs it inside the whole process. Now let's use another example. We mentioned that energy can be another problem, and it may be that, for example, it can't break down lactose. So there's a bacteria that has a mutation on the gene that codes for lactase, and therefore it can break down lactose. This is essentially another example. Let's see how we will denotate this. So lac negative, right out the bat, we know that this bacteria cannot grow in just with lactose as energy source. This negative indicates that it cannot grow with just lactose. And lactase plus indicates that this bacteria can survive just on lactose. It has the ability to metabolize lactose for its purposes. The negative, on the other hand, is just showing that, okay, you have a negative. That means that you need, let's say, any other kind of energy source to make up for it. Now you should probably know the overall terminology you would need in your first bio lab. But this is essential because eventually you will be understanding, as we talk about bacteria, you'll understand the entire mechanism behind their growth and their function. So good on you.